Welcome to this first part of the molecular modeling session. And today we will talk about molecular visualization. So my name is KnutTeigen. I'm a professor in biomedicine at the Department of Biomedicine here in Bergen. And I'm interested in proteins and protein structures and how small molecules bind to proteins and how they change the conformations of proteins and change the function of proteins and how you can develop small molecules into drugs that modify protein function. So that's sort of my interest. But today we will talk about visualization, how you can use computers to visualize molecules, small molecules and larger molecules like proteins, and why I think this is important also to show structures because looking at structures you can start to understand something about their function, and also you can start making hypotheses about what these proteins do or small molecules do. So very often it's important to have a model that you can use to predict function. So, but before we start with the different softwares, I want to show you 3 different softwares that we use a lot in my group to look at structures. And so I'll have a small demonstration of those softwares. But before we start with that, I want to talk a little bit about visualization and how important it is in general in science, but maybe particularly in biomedicine and medicine in general. So these drawings are from Leonardo da Vinci, made over 500 years ago, and it's the anatomy of the human body. So with these drawings, he shows how the different muscles are connected to the bones and to the joints and how they function. So these structures are not easily accessible. You have to dissect the cadaver to actually see these structures. So that's what he did, but he made really beautiful drawings of this. So it would be so then it's more easily accessible for anyone that to just open a book and look at these. And based on this visualization of the human body and internal organs and the muscles and the joints, you can start to understand the function of muscles, how they work and how they function together to move the body in different directions. So based on this visualization or drawings, you could start to interpret or understand the function more of the human body, and especially anatomy. Um, and it's the same today in medicine. We use a lot of different models. For example, of the human heart, it might be difficult to understand just inside your head how the heart works, how the blood comes into one chamber and then is pumped out to the rest of the body. But if you have a physical model you can play around with, it's more easy, at least for most people, to then understand how this works and how things are connected, and then this might give you new questions you can ask and to understand really the system. And same thing, for example, with the kidneys or with the brain, you have different structures, and to really understand how these work together, it's really nice to have a physical model you can play around with. And these structural models, they give you some sort of understanding of the function of these different organs. And it's kind of the same with molecules. So this is one of the first models of a molecule that is known. It's from Wilhelm von Hofmann. This model is still on display at the Imperial College in London. And if we look at this model now, we see that it's not correct because it's a methane molecule. And so you have 1 carbon with 4 hydrogen atoms around, and it shouldn't be flat like this model shows. But still, it was an important model at the time because it said something about how atoms are connected in a molecule, And with this model, you could start asking questions about the function of molecules and how they're sort of connected. And so this is quite a long time ago, and we have more sophisticated models of different molecules, and maybe one of the most important molecular structures is the double helix of DNA that was discovered back in the 50s. And Watson and Crick, I guess, got most of the credit, but there was also a person, Rosalind Franklin, that did a lot of the work and maybe most of the work, and also solved the X-ray structure of this. So she should maybe have even more credit than these two guys, but still, it's a really important structure that showed the double helix of DNA and how the different bases, the base pairing in the DNA. So this opened up a whole new area of science. So in molecular bio or in genetics, and for example, we wouldn't have the CRISPR techniques now if we didn't really understand the structure of DNA. So the structure of DNA was really important to understand the function of DNA. So based on this, you could start to understand how you can manipulate DNA and yeah, get a deeper understanding of how inheritance work. And it's sort of the same with the models that were made 500 years ago about the human body. You sort of started to using these models, you could understand the human body on a whole different level with the muscles and joints and how everything is connected. And it's kind of the same with the model of DNA that we got back in the 50s, where you could then really start understanding how DNA works and how everything is connected and how mutations influence um, yeah, the structure and how it what's really going on. So having knowledge of the structures, you could really start saying something about the function. And it's the same thing with developing drugs and small molecules that are supposed to bind to different receptors, usually a protein. So to really understand how these work, you need to have knowledge about the structure of the complex. So for example, with antibiotic drugs, to understand how they work, you need to have a understanding of how they bind covalently in the binding site of the bacterial enzyme and how they inhibit this. And if you want to make analogs of or make new antibiotics, you need to understand or have an understanding of the binding site, what it looks like, and what sort of modifications you can do to these different molecules to maybe make them bind even tighter, and in that way make an even more specific drug molecule. So having knowledge about the structure gives you some understanding of the function, and you can use that to, for example, develop new drugs. So yeah, one way to talk about drug design, I just wanted to show you this poster back. That's from 1885. And this was more or less how drugs were not developed but discovered in former days, where they found some compound that usually a biological compound that's found in nature that had some sort of biological effect. And in this case, it's cocaine that's given as toothache drops to children with problems with their teeth or having pain, because cocaine has a small anesthetic effect as well. But of course, now we know that cocaine is absolutely nothing you should be distributing to kids with pain in their teeth. Cocaine has a lot of side effects that you really don't want to have in kids, like violent behavior and all this. And yeah, so yeah, but this was just to show you that in former days, it was usually that you had some compound that had some biological effect, and then you use that as some sort of drug. But nowadays, you really need to know the the structure of things and how what it binds to and how it activates different receptors, for example. So, and well, you have to go through all these phase clinical trials and all this to get the drug to the market, but that's a completely different story. But you really need to know the structure as well at the molecular or atomic level of how things work usually. So with that kind of long introduction, I want to show you some different types of software that we use. One type of software that we use a lot to look at small molecules and just in 2 dimensions, and to look at small molecules, and then also some software that we use to visualize more larger molecules where it doesn't make sense to show every atom explicitly, but for example, for a protein, you want to show the secondary structures in the protein, and then you need or usually use a different kind of software for doing that. So with that, I just want to show you one software for looking at small molecules, and there are several different ones, but the one I want to show you is for free, so you can download it and use it for whatever you want. And that software is called BioVia Draw. So I'll just show you a short, just some things you can do in BioVia. So it's fairly simple and easy to use. So you have this canvas where you can draw structures. So for example, you have these templates that you can use. So for example, I want to make some rings here, some 6-membered rings. And then I want to make a 5-membered ring here, and then maybe I want to add some bonds. I want an extra bond here, and let's see, I want this one the other way. Let's see, I want one here, and I want to add something here, and then here, for example, and then I want this to be an oxygen. Let's see. Let's see, I want to have a hydrogen on this one. So if I do like that, it should work. And then also I want to make this into a oxygen. Let's see here. Oops. Let's see if I do like this, then I get an oxygen, and I want a double bond here, for example, and I also want this to be a double bond. So this is just a fairly easy way of sketching a molecule. So maybe some of you recognize this one, but let's see if I can find this one. So just make this a little bit bigger. If you haven't figured out what this is testosterone. So it's fairly easy to just draw different structures that you want. And there's one nice thing here. You can have calculator. So this calculator gives you some properties of the molecule that you just made. So in this case, it gives you, for example, how many hydrogen bonds, acceptor and donors you have, the composition of your molecule, and the mass, and the number of stereocenters, and it also gives you the hydrophobicity of your compound, which can be interesting, and also the polar surface area, which you might be interested in if you're developing a drug. It tells you something about the distribution of this molecule, and how if it binds to plasma proteins and all this. So it gives you some sort of indication on the properties of your molecule. And another very nice thing too is that if you draw such a structure, it sometimes is difficult to figure out the UPAC name. If it's been a long time since you had organic chemistry and you don't have any friends who are organic chemists, you can go here and you say generate text from structure, and you want the UPAC name. So then it pops up here. So then you have the correct, usually it's the correct, the software is quite good at this to make the UPAC name of the structure that you just made. So that can be quite useful. I'll just make a new one, and just to show you that you have also different templates here. So for example, if I want to have an amino acid, here we have all the different amino acids. And for example, I want a phenylalanine. So I just click here and then I get the phenylalanine. So then I want to know something about this structure. So I'm going to have the calculator open. Then I can say that it should calculate as I draw. So if you look at this, it has a certain hydrophobicity and a number of hydrogen bond acceptors and donors. And then I add a hydroxyl group here. And then you see that you have more possibilities to make hydrogen bonds and accept hydrogen, or hydrogen bond donors and acceptors. And it also becomes more hydrophilic, your molecule, which is not too surprising. And also the polar surface area changes. So with this modification, it's not so surprising, but sometimes when you work with small molecules, it can be nice to just have this calculator that gives you some information about the properties of your molecule. And there is also other nice features here. So for example, if you go to generate structure from text, you can have different input, for example, a smiles string. And often when you work, if you have a database of small molecules, often when you work with them computationally, they are just given as a smiles string, and a smile string is just a string with letters that tells you which atoms are in your molecule and the connectivity between them. So it's just a very simple way of defining in one string molecule. So you can add that here, and if you add the smile strings, string, you will get the structure. And in the same way, if you have the UPAC name, if you read a paper and they talk about some compound they made and they give just a UPAC name, you can add the UPAC name here, and it gives you the structure. And it also has a lot of common names or historical names of compounds. So for example, cholesterol, even if it's not the UPAC name, it understands that. So then you get cholesterol with all the correct chirality and all that. And for those of you interested in biochemistry, you can see that testosterone is made from cholesterol. And then, of course, if you just move this a little bit, if you want to have other molecules made, so for example, you're interested in, let's say, cocaine, then you have the structure of cocaine. So in this way, it's very easy to get the structures and manipulate them and modify them if you need to. Instead of, for example, going to Wikipedia or Google to look for a structure and download some sort of graphic file. If you want to use it in a paper or in your thesis, it's much easier just actually to just draw them or use the structure, generate structure from text here. And then you get them in the same format, and then you can save them as image files like PNG or JPEG or whatever you want, and put them into Word or PowerPoint or some other software that you want to use. So this is I think it's very useful. And if you want to make more sophisticated drawings, maybe you want to have some sort of membrane or maybe micelle, and you want to maybe you have some sort of receptor, you can add receptors, and yeah, all these kind of all these different templates that you can use for example, an enzyme, or so you can play around with this and draw small molecules. So you can make quite sophisticated figures in this software. Yes, so I think that was more or less what I wanted to show you with uh, with BioVia Draw. So the very nice thing is that it's for free, and you can go online and you can download it from this webpage, or you can just Google BioVia Draw and you will find it. So it's very nice for drawing 2D structures, and then you can export that into and use it in some other software that you have. And as we saw, you can use this to get some idea about the function of your molecules by this calculator. And yeah, so that can be quite useful if you work with small molecules. But if you work with larger molecules like proteins or DNA, it's often not very useful to explicitly show every single atom of your structure. You want to show, for example, the secondary structures of proteins and or a simplified way of visualizing it. So then PYMOL is something that's very useful for that. PYMOL, it used to be open source and for free, but then it was bought by Schrödinger. So now there is a license for PYMOL, but still there is, it's possible to get the code because it's an open source software. So you can, if you work with, if you use Linux machines, you have to compile it because you get the source code of the software. So you can compile it for Linux, for example, but also for Windows. You can also have a educational version of PyMol, but the link is not so easy to find, but you can find it. It's here. If you go to pymol.org/edu, you can get the educational version of PyMol that's for free. I think there are some limitations, and you're not really supposed to use it for publications. But yeah, if you are at the, I think our department of biomedicine has a license for PyMol, and if you're in a group that work with structures, usually they have a license for PyMol because it's not so expensive and it's really useful. So I just want to show you very briefly what PyMol looks like and what you can use it for. So this is PyMol. So it was originally developed for Linux. So it's command based. So there is a lot of commands that you can write, and it might be a little bit confusing in the beginning. It takes a little bit of time to get used to, but you also have all these drop-down menus that you can use. So it's more or less like a Windows software. And they have some demos. I just want to show you some of them. So there are different ways of visualizing. This is just a very short peptide. So you can visualize every single atom of your peptide, like with lines or sticks or van der Waals radius or spheres, and also the surface of your peptide. And you can also visualize the secondary structure, like ribbons and cartoons. So this is just a short antiparallel beta sheet. So there are different ways of visualizing. So if you look at this, then you can look. So you can also have sort of the surface of your peptide, and then So if it's transparent, you can see the actual amino acids beneath. So another thing that's kind of useful if you show the electrostatic onto the electrostatic potential onto the surface of your molecule, you can see that you can see sort of the shape of your molecule, and you can see that you have some red and blue parts. The blue parts are positively charged and red are negatively charged. So if this molecule or is supposed to bind to some binding pocket, there should be some complementarity in the binding pocket. So you should have some sort of positive charge in this area and negative in this to complement the positive and negative charge of your molecule. So this might give you some understanding of the properties of your small molecules if you visualize the electrostatic potential onto the surface. What else did I want to show you? Yeah, so with this, you can also make videos. So you can make short animations very easily in this. You have this movie function where you can add rotations and different if you have different molecules that you want to come together, you can put that in here in different frames, and then you can have this as a video. So in this case, it's HIV protease, where with an inhibitor in the binding pocket. And when you're happy with what your video looks like, you can go to File and export the movie as different formats, and then you can put them on your web page, or you can have them in PowerPoint, or yeah, whatever you want to do with the video. So that can be quite useful. And you also have this ray tracing capability where you have if you have some sort of structure, sometimes it can be difficult to sort of get a 3D impression of what it looks like. But with the ray tracing, you sort of get you have these shadow effects, so you get more of an impression of the 3D structure. Let's see, and also, yeah, so you can visualize proteins by secondary structure, and then you can color them the way you want. For example, if I want to color this by secondary structure and I want the helices to be red, beta sheets to be yellow, and loops to be green, so then you get sort of an impression of the secondary structure of your protein, and maybe you can say something about the function. In this case, it's a transmembrane protein, so it seems like this helix goes through the membrane, and then you have a domain on each side of the membrane. Yes, so I think that was more or less what I wanted to show you with this. I'll just remove everything, and I want to just read in a PDB file. So we will come back to the protein data bank, but this software is connected to the protein data bank, so I can type in just a command so it's a 4-letter code that is a code for the coordinates of a specific structure in the protein data bank. So if I type in that, I get the structure here, and this is protein kinase A, the regulatory domain with cyclic AMP bound here and here. So I have 2 cyclic AMP molecules bound to the protein kinase protein. So so then you can start looking at the structure, and then you have some presets that are kind of useful. So for example, if I go to actions and I want to go to preset and I want to have a publication, if you click that, then you get publication quality sort of structure where you can see the ligands and the protein, and then if you want to, you can type ray, and then it ray traces this. So then you get sort of the 3D effects of with shadows and the transparent background. So then you can save this as a graphics file and open it in Word or PowerPoint or whatever, and to show it. And there is another really nice preset here. So you go to preset and you want to look at the ligand sites, and I want to have solid surface around the ligands. So now you can see the binding pockets of the cyclic AMP. So if I select this one, let's see, sorry, I'll select cyclic AMP, and I want to zoom in on that So then we see the binding pocket of one of the two cyclic AMP molecules. And then you can see you get dots here to different amino acids that are involved in interactions with cyclic AMP. So you can, for example, you can click on this one and it gives you, this is alanine 210. So then you can start, and it seems like you have some sort of stacking interaction here. So if I click on this one, this is tryptophan 260. So this gives you some sort of information about how the ligand binds to the protein. So there is a lot of other things you can do with PYMOL. I won't go into too much detail. Just show you the software, and if you're interested, download it and play around with PYMOL. And the very nice thing with PYMOL is that there is a very active community using PYMOL, and there's a PYMOL wiki page where you can go and find answers to any type of question you can think of and probably not think of, and examples of how to make different types of figures or graphics. And and there are also tutorials if you want to get to know the software. And this PYMOL wiki page is really useful. I just want to show you one last software to look at structures, and it's also from BioVia. But this is for looking at larger molecules, like with PYMOL. But I think this from BioVia is easier to use than PYMOL is. PYMOL is really powerful, and you can do anything and whatever you want, especially if you start making scripts, and you can make extremely nice graphics with all this ray tracing and all those fancy things. But if you just want to look at the structure and you're not too familiar with typing commands and using PYMOL, I would recommend using Discovery Studio from BioVia, which is really easy to use. The disadvantage maybe is that the graphics is, well, it's good enough to have in a publication, but if you want to make really impressive graphics, then maybe PYMOL is better. So I'll just show you very briefly with Discovery Studio what you can do, or at least some of the things you can do. So here I'll just open the same structure. So this is again protein kinase A with two cyclic AMP molecules bound. So one nice thing you have here in Discovery Studio is that it's very easy to find the different amino acids that you're interested in. So for example, if you read a paper about protein kinase A, and they say that they mutated, let's see, tryptophan number 260. So you find it here. And then since this is selected now, I can go to display style and say I want to show this as sticks. And then I zoom in on that. And then you can see that this amino acid makes stacking interactions, or at least it's very close proximity to the cyclic A. This is the cyclic AMP molecule. So if you mutate this, then you would expect that you lose affinity for cyclic AMP. So if you read the paper about structures, I would really recommend to download Discovery Studio and just open up the structure. So you can also go here, open URL and type in the four-letter code of the structure, and then it gets it for you directly from the protein data bank. And then you can start playing around and look at what's going on. So I'll just remove this again and show you one other feature that's kind of nice. If you select the cyclic AMP molecule, you can go to Edit, Select, and then you want to add amino acids in a radius within, let's say, 5 angstrom. So I do that, and I want to have the complete amino acids. So now all the amino acids that are within 5 angstrom of cyclic AMP are selected. So then I want to display these as sticks. So then suddenly you see all the amino acids that are in close proximity to cyclic AMP. So you see that you get the tryptophan 260 that we talked about. And then there seem to be some sort of interaction here with this oxygen down to this glutamic acid. So if I double click on this, you see that, oops, if this is glutamic acid 200. So this gives you some information about the binding site, which then can be useful. And there is one really nice tool. So let's see, I'll just take away that. And then there are these tools, and you have receptor ligand interactions. So I want to define the ligand. So I go to cyclic AMP and I define that as the ligand. And then I can click ligand interactions. And then it actually shows all the interactions that you have, and it's color coded. So you have some stacking interactions, you have some hydrogen bonds, and then so then you get a lot of information just by doing a few clicks here. So then you can select this one, and this is alanine 211. So it gives you information about how this compound binds to the protein. Then you can also click on this, show 2D diagram, and then you get cyclic AMP here, and then you get all the different amino acids that are interacting. Color coded again, so green is conventional hydrogen bonds, and then you have some sort of stacking interactions here. So this is really nice and useful, and so you can also add the receptor if you want to have the whole receptor, and I guess you can also have the surface, so you can have the charge of the binding pocket. So then you can see it's color coded by charge, so it's red for negatively charged pocket. So so then you get a lot of different information just by clicking around here. So so that's what I wanted to show you about BioVia Draw, and it's really powerful and extremely easy to use. You can do everything that you can do, I guess, at least almost everything, or I guess everything that you can do in BioVia Discovery Studio, you can do in PYMOL, but it's a little bit more difficult to get it done because you have to write all these scripts, but then you can do a million more things in PYMOL if you really know the software, and you can make extremely nice graphics in PYMOL if you spend some time to get to know it. Yes, and there is one nice thing with BioVia with Discovery Studio, that there is also a plugin for PowerPoint. I now just realized that I haven't installed that plugin on this computer. But if I had, you could have an ActiveX plugin here where you can actually modify and you can rotate your molecule in PowerPoint and to turn it around and click on different amino acids if you want to. For example, if you want to impress during your master thesis presentation, you can implement Discovery Studio in PowerPoint, so you can play around with the model while you're showing it in PowerPoint, which can be kind of cool. So with that, we've looked at some different types of software. We've got the BioVia Draw to look at 2D structures of small molecules, where you can use the calculator to get some properties of the molecule that you have. And you can also have this to generate structure from text, where you can type in a UPAC name or just a common name of a compound, and then you get the structure. And then you can save that as a JPEG, for example, or a PNG file, and put it into a paper or your thesis or wherever you want to have it. And then we looked at the PYMOL. for rendering molecules in 3D. And you can have this really nice graphics that you can make with PYMOL, and you can also make videos that you can put on web pages if you want to. But if you just want to look at structures and figure out how, for example, a ligand binds to the active site, I would recommend using BioVia Discovery Studio, which is really easy to use, and you can manipulate your model and clicking at different amino acids to get to visualize it. So Discovery Studio is really easy to use. But if you're used to PyMol, it's much more powerful, and you have much more possibilities in PyMol. Yes. And I just want to show you some more sophisticated or strange ways of visualizing. You have these caves where you can go into your model. So so it's so you have screen in the ceiling and the floor and the walls, and then there is a detector in the roof that detects where you are, and you have these 3D glasses on, so it feels like you're inside the protein. So you can walk around and look at different amino acids, and if you have a ligand bound in there, then you can study the interactions, and you even have versions where you have like a joystick, and you can actually feel how much force you need to apply to remove a ligand from the binding site, well, calculated by the computer, if you have a strong enough computer running all this. But I'm not quite sure how useful it is. It's kind of impressive, and well, it's an interesting tool that they have some at the university hospital. I think they mainly use them for MR pictures or models where you can, for example, have a picture of a brain where you can walk around, and you can, before you do surgery, and then you can look at the model in 3D and yeah, plan for the operation. And of course, you can also install games on these. You can have monsters running after you, and you can have all these different weapons where you can shoot, and you can probably spend a full day playing around with these caves. Yeah, so I just wanted to show you that. But and you also have these more sophisticated ways of ray tracing molecules, and now we're moving more maybe from biomedicine and more into art science or art field. So you have, for example, you can have like a protein that's in the water, and you have all these nice 3D effects with ray tracing. You can use PowerRay for doing that. So if you go to powerray.org, you can also download that for free and play around with coordinates to visualize them and ray trace them. So you can get these really strong 3D effects, and you can make it even more sophisticated. So you can have a protein in the sunset, and you can also have melting DNA. This yeah, is maybe a bit over the top, but still, I think it has something to do with if you're at the conference and you present a poster and you have something like this on your poster, you get a lot of attention. So so it's kind of a way of attracting attention, I guess. And if you publish a paper, very often the journal will ask you if you have something that they can put on the cover of their issue, and if you send them something like this, I'm quite sure they want to put it on the cover, and that's kind of nice to have on your CV. If you send a paper to Science and they put you on the cover of Science magazine, that's kind of cool to put on your CV. So it could make sense to make all these sort of sophisticated ways of visualizing molecules, but I guess these last ones are more for those that are especially interested in that. And it takes quite a lot of time, but if you're interested in that, then of course go ahead. So yeah, with that was more or less what I wanted to say about visualization, and I think it's really useful to visualize molecules, to understand something about function, and I think it's really important to to use these tools if you work with molecular structures, because it can give you maybe not necessarily a lot of answers, but it can give you really good questions that you can go in the lab and test. Okay, so with that, I think we're done with the visualization part of this, and we'll talk a little bit more about molecular modeling when we actually meet in the auditorium. So with that, thanks, and see you soon.